Hello everyone. In continuation to our topic research methods, now we will see one of the experimental design which is called completely randomized design or abbreviated as CRD. CRD is a type of experimental design where participants or any units are randomly assigned to treatment groups. There are no pre-existing groups or blocking variables. The goal is to compare the means of two or more treatment groups, generally more than two. I can say means of more than two treatment groups. Key features of completely randomized design. One is random assignment of participants. Another one is one independent variable, factor. Variable is also called as factor, which with multiple levels. No blocking or stratification. The objectives of completely randomized design are comparison, means we can compare the effects of two or more treatments, that is independent variable, on a response variable, that is dependent variable. Inference, to make inferences about the population based on the sample data. It also minimizes the bias, means to minimize systematic bias by randomly assigning participants to treatment groups. Maximization of precision is achieved by ensuring that every participant has an equal chance of being assigned to any of the treatment group. Testing of hypothesis is possible about the effects of treatment on the response variable, means the dependent variable. So in a CRD, the objective is to determine whether there are significant differences between the treatment groups. Let us understand this through an example. Suppose we want to compare the effects of three different fertilizers, A, B, and C, on plant growth. Let us say we have 30 identical plants and want to randomly assign them to these three fertilizer groups. What are the steps to follow? Step 1, randomly assign the 30 plants to three different fertilizer groups, like 10 plants per group. Then apply fertilizer A to the group 1, fertilizer B to the second group, fertilizer C to the third group. Then measure and record the plant growth. For example, it can be height or weight, generally height after a set period. The resulting data may look like this. By using fertilizer A, in that group, you get height in centimeter as 10, 12, 8, 11, 9, 13, 10, 12, 9, 11, like that. Similarly, for fertilizer B, the second group will have readings like, may have readings like 12, 9, 14, 10, 13, 11, 12, 10, 14, 9. Similarly, to the third group, where you had used fertilizer C, you may be getting a reading like 9, 11, 10, 12, 8, 13, 9, 11, 10, and 12. So this design allows us to compare the mean plant growth across the three fertilizer groups, ensuring that any differences are due to the fertilizers and no other factors. Advantages of completely randomized design are simple to design and implement. It minimizes bias. It allows for easy analysis. But there are disadvantages also, because it may not control all sources of variation and may not be suitable for large experiments. For doing analysis, we use what is called as ANOVA, or analysis of variance, which is a one-way ANOVA, what we will be doing to analyze a completely randomized design. The next design is the Randomized Block Design, abbreviated as RBD. It is a type of experimental design used to reduce the impact of extraneous variables on the outcome. It is commonly used in agriculture, medicine, and social sciences. Let us understand this with an example. Suppose we want to compare the effects of three different fertilizers, A, B, and C, on wheat yield. We have 12 plots of land but the soil quality varies across the plots. To minimize the impact of soil quality, we divide the plots into 4 blocks, each containing 3 plots with a similar quality as follows. This is first block, second block, third block, fourth block. In this block, all the plots will have a similar soil quality. In this also, the plots are homogeneous as far as soil quality is concerned. Here also you find a homogeneity. Here also you find a homogeneity. You can also call this as stratification or blocking. In each block, the 3 fertilizers are randomly assigned to the plots. You may notice here A, B, C in each of the plot. Here also ABC, here also ABC, here also ABC, but the order is different. In each block, the 3 fertilizers are randomly assigned. That is why the order is different to the plots. This ensures that the effect of soil quality is minimized and we can compare the fertilizers more accurately. By using a randomized block design, we can reduce the impact of extraneous variables, that is soil quality, increase the precision of our estimates, and compare the fertilizers more accurately. So this design is particularly useful when there are multiple sources of variation and we want to control for one or more of them. What are the key features of this design? Blocking is the key feature, means division of the experimental units into homogeneous groups or blocks to reduce variability. That is what we did here. Block 1 is being divided into 3 different plots. Or we can say the entire thing is being divided into different blocks. Randomization. Random assignment of treatments to plots within each block. And then replication. Each treatment is replicated in every block. Control of extraneous variables. Blocking helps us to control for extraneous variables that might affect the outcome. It leads to an increased precision because RBD increases the precision of estimates by reducing the variability. Improved accuracy by controlling for extraneous variables. Randomized block design improves the accuracy of treatment comparisons. Flexibility, RBD can be used for various experimental designs, including factorial designs. Efficient use of resources, RBD can reduce the number of experimental units required. Analysis of variance is typically used for analyzing RBD data to compare treatment means. In this, assumptions are RBD assumes that the blocks are homogeneous and that the treatments are assigned randomly within each block. By incorporating these features, RBD provides a robust and efficient design for comparing treatments while controlling for extraneous variables. What are the advantages of RBD? It reduces variability because blocking reduces variability due to extraneous variables, making treatment comparisons more precise. Further, it increases precision of the estimates by controlling for extraneous variables. It improves accuracy because by reducing variability, RBD improves the accuracy of treatment comparisons. It enhances validity. It helps ensure that treatment effects are due to the treatments themselves and not due to the extraneous variables. Efficient use of resources is possible because RBD can reduce the number of experimental units required. Flexibility. RBD can be used for various experimental designs, including factorial designs. Easy to analyze because RBD data is typically analyzed using analysis of variance, a straightforward. Only difference is here it will be a two-way ANOVA, a straightforward and well-established method. It reduces bias because randomization within blocks reduces bias and ensures that treatment assignments are unbiased. It accounts for covariates. RBD can account for covariates, means extraneous variables that might affect the outcome. Wide applicability, RBD is suitable for various fields, including agriculture, medicine, social sciences, and extra, and more. Disadvantages, limited flexibility. RBD is less flexible compared to other designs as it requires a fixed number of treatments and blocks. Difficulty in adding new treatments. Adding new treatments to an existing RBD can be challenging. You may have to restart from the beginning. Inefficient for large number of treatments. RBD can become impractical for a large number of treatments as the number of blocks and plots increases rapidly. You need homogeneous blocks. RBD assumes homogeneous blocks, which can be difficult to achieve in practicality in practice. Sensitive to block treatment interaction, RBD assumes no interaction between blocks and treatments, which may not be true always. Analysis can be complex. While ANOVA is commonly used, the analysis of RBD data can become complex, especially with multiple factors. It requires careful blocking. Blocking must be done carefully to ensure that the blocks are truly homogeneous. Limited ability to control for multiple extraneous variables. RBD can only control for one extraneous variable, that is the blocking variable. It may be suitable for small experiments. RBD may not be the best choice for small experiments as it requires, here I should have said may not be, as it requires a number of blocks and plots. It can be time consuming and resource intensive. RBD can be time consuming and resource intensive, especially for large experiments.